Welcome to the deep dive. This is where we take your sources, could be articles, studies, notes, whatever you send us, and we really get into them.
Yeah, we pull out the key insights, the stuff you need to know, minus all the fluff, trying to make complex stuff, understandable.
And maybe even a little entertaining along the way.
We try.
Okay, so today's topic is fascinating. It's C60 fullerene. Maybe you've heard of it as carbon 60.
Or Buckminster fullerene, if you want the full name, big Nobel Prize winner.
Right, and it's got this buzz around it, especially as like a potential health supplement. But as usual, when we dig in, it gets complicated.
It really does. And for this dive, we've got a great mix of sources. There's this personal review, like a blog post, from someone actually taking C60 oil.
Okay, real world experience.
Exactly. And then we have several pretty dense scientific papers looking at lab results, safety, how it works biologically.
So the anecdote and the data side by side, that's perfect for a deep dive.
Mm.
And actually, we really need to thank redlighttherapy.rehab for kicking this off. They sent us these sources to review.
Big thanks to them. It's this combo of user perspective and hard science that really lets us, unpack things properly.
So our mission today, to figure out what this collection of stuff tells us about C60, from the benefits someone feels they're getting,
to what the lab studies say about potential effects, both good and maybe not so good, and the risks.
Okay, let's get into it. First things first. What is C60, basically?
Okay, so imagine a molecule that looks like a tiny, tiny soccer ball, or like a cage. Carbon. Exactly 60 carbon atoms all arranged in this perfect sphere. That's C60.
Buckyball.
Right, the buckyball. And it's not like ancient knowledge, is it? Pretty modern discovery.
Relatively, yeah. Discovered in '85, got the Nobel Prize in chemistry for it in '96. So it's definitely real, recognized science.
Okay, and the health supplement angle, where does that stem from?
Well, one of the sources talks about its potential as a really powerful antioxidant.
Antioxidant, okay, like vitamin C. Sort of, but potentially different. The idea is its unique cage structure lets it grab onto multiple free radicals, those damaging molecules in the body, without getting used up itself.
Oh, interesting. So it keeps working.
That's the theory. Unlike typical antioxidants that sacrifice themselves, C60 might be able to neutralize many radicals over time. That potential antioxidant power is really the hook for a lot of the health interest.
Okay, antioxidant power. That leads us to probably the most famous study mentioned in the blog post, right? The body study from 2012 with rats.
Ah, yes, the body study. It's almost legendary in C60 circles. And honestly, the results were completely unexpected.
How so? What were they actually looking for?
They were looking for toxicity. They weren't trying to find a miracle cure or anything.
Wait, toxicity? Why do they think it would be toxic?
Well, because C60's structure has some similarities to benzene.
Ah, okay. Benzene's not good stuff.
Definitely not. So they were giving C60 dissolved in olive oil to rats orally to see if it caused harm, maybe cancer or other problems long term. They were basically doing a safety check.
And instead of finding problems...
Instead, they found something pretty shocking. At the dose they used, the C60 in olive oil wasn't toxic at all, and it massively extended the rat's lifespan.
Massively. How much are we talking? The source gave numbers.
Yeah, the numbers are wild. The control rats, the ones just getting olive oil, lived about 22 months on average.
Okay, typical for these rats.
Right. The rats getting the C60 in olive oil, their median lifespan jumped to 42 months.
42? That's almost double, a 90% increase.
90%. It's an incredible figure. Really turned heads.
Wow. That does sound unbelievable. Almost too good to be true from one study.
Well, yeah. And there's another weird detail. They actually stopped giving the rats C60 around month 17.
So they stopped the treatment halfway through the expected lifespan.
But the benefits kept going. That increased lifespan just continued. One of the C60 rats apparently lived to 66 months.
66 months, even after stopping the C60 way earlier.
Correct. The effects seem to persist. Now, you got to layer on the caveats, which the blog post itself does mention.
Right. Important context.
Super important. It's one study. It was a small study. And crucially, it was in rats, specifically Sprague-Dawley rats.
Not humans.
Definitely not humans.
Yeah.
And translating findings from rats to people is always a huge leap. Lots of unknowns.
Okay, so a potentially groundbreaking finding in rats, but needs way more investigation. Which brings us to the personal account, blog post, someone actually using it.
Yeah, this shifts the perspective. It's someone's lived experience, taking C60 as part of their whole health routine. They mention a specific diet, red light therapy too.
Right, so it's not isolated, it's C60 plus other things.
Exactly. Makes it hard to pin down cause and effect, but still interesting to hear their perceptions.
So what did they feel they were getting from the C60 oil?
They listed a few key things. First, that potent antioxidant power. We talked about the idea it neutralizes radicals without degrading.
Oh.
They also mentioned feeling it supported cardiovascular health, maybe helped with triglycerides, blood pressure, though they did note that overlaps with their diet changes too.
Yeah, that's always a challenge with personal reports. Hard to isolate variables.
Totally. They also felt a kind of mitochondrial boost, like more stable energy throughout the day.
Energy boost. Okay, a common goal.
Mm-hmm. And better cognitive clarity. Said it helped get rid of brain fog.
Energy and focus, big ones for many people.
Yeah. And finally, they mentioned inflammation control. Felt it gave them an extra edge, especially recovering from workouts or stress.
Interesting. Did they talk about how they take it or quality?
Oh, yeah. They emphasized that. Taking it dissolved in extra virgin olive oil. Seems C60 needs a good fat carrier for absorption.
Right.
And they really stressed getting high-quality stuff. Lab tested, purity over 99.9%, properly dissolved, no visible bits or sludge, and from a transparent brand without fillers.
Makes total sense. If you're putting something like this in your body, you want to know exactly what it is.
Absolutely critical. Okay, so we have this intriguing rat study, this positive personal report. But now we need to balance that with the other science.
Yeah, the study's looking specifically at potential risks and mechanisms. This is where it gets maybe a bit more complicated.
Yeah, definitely more complex. Because while we've heard potential good stuff, these other controlled experiments highlight potential problems in lab settings, things related to the heart, and how C60 actually behaves chemically.
Okay, let's start with that rat study focused on heart injury. What was that about?
Right, another study using Sprague Dolly rats. This one looked at what happens if the heart muscle loses blood flow and then gets it back. It's called ischemia-reperfusion injury. Think heart attack recovery.
Okay, a stressful event for the heart.
Exactly. And they gave the rats C60 in two different ways, either through the lungs, like inhaling particles.
Intertrail, right.
Yep. Or injected directly into the veins, intravenous.
And an impact on heart damage.
This is the key finding. Both methods of C60 exposure made the heart damage worse after that ischemia-perfusion event. The area of dead heart tissue, the infarct size, was bigger in the C60 exposed rats.
Whoa. So, opposite of the longevity finding, in this specific injury model, C60 seemed to make the heart more vulnerable.
That's what the study suggests, yeah. A really stark contrast. It just shows how context-dependent these effects might be.
Did they look at anything else, like blood vessel function?
They did.
Yeah.
They looked at the coronary arteries, the ones feeding the heart muscle, how they constricted or relaxed.
More complexity.
Definitely. It depended on how the C60 was given and even the sex of the rat.
Okay.
So when C60 went in via the lungs, the IT route, male rats tended to have their vessels tighten up more in response to certain signals, potentially restricting blood flow.
And if it was injected, the IV route.
For male rats, IVC60 seemed to impair the vessel's ability to relax properly in response to other signals. Plus, they saw higher levels of certain inflammation markers in the blood with the IV route.
Huh. So the delivery method really changed the specific cardiovascular effect.
That's wild. It is. And there were gender differences, too. Female rats seemed more susceptible to the heart injury after lung exposure compared to IV exposure. The opposite trend was sort of seen in males.
Really? Why?
The study suggested maybe differences in inflammation or how their vessels reacted. The females with lung exposure had a weaker relaxation response to a key signal, possibly contributing to the bigger infarcts.
Man, okay. So C60's effect on the heart and vessels seems really intricate. Depends on how it gets in, maybe your sex, the specific situation.
Precisely. The researchers theorized links to inflammation, maybe issues with blood clotting or breakdown, creating no reflow zones after injury. But interestingly, they didn't see major lung inflammation or obvious cell toxicity at the dose used. The effects seemed quite specific to the cardiovascular system under stress.
Okay, complicated picture for the rats. What about the zebrafish study? What was the focus there?
That one went after the mechanism. Why might C60 be toxic sometimes? They used tiny zebrafish embryos.
And the verdict?
Their main finding points strongly towards C60 acting as a powerful oxidant, causing oxidative stress.
Hold on. Oxidant? I thought the big selling point was antioxidant. How can it be both?
That's the million-dollar question, isn't it? And where things get super counterintuitive, this study found C60 caused oxidative stress in these embryos. And here's a key twist. Light mattered.
Light. Like sunlight.
Just regular light. They found the toxic effects cell death, Malformations were much, much lower if the zebrafish were kept in the dark during C60 exposure.
So light exposure could activate C60, making it cause oxidative damage.
That's the strong implication from this study, photoactivation leading to oxidative stress. They also tested the role of the embryo's own defenses.
Like its internal antioxidant system.
Exactly. They used chemicals to block glutathione production. Glutathione is a major natural antioxidant. When they did that, the embryos became way more sensitive to C60. Lower doses caused harm.
Okay, so the body's own antioxidant status is critical in how it handles C60.
Seems so. And the flip side, adding extra antioxidants like NAC or vitamin C protected the zebrafish. It reduced mortality, deformities, suggesting you can buffer against this oxidative effect.
So this zebrafish work paints C60, at least the unmodified form under light, as a potential powerful oxidant driving toxicity via oxidative stress and influenced by antioxidant levels. That definitely complicates the simple antioxidant supplement narrative.
It hugely complicates it. Okay, one more study to touch on the mouse study. This one looked at mice prone to atherosclerosis, so they already had some vascular vulnerability.
Like studying it in a population perhaps more at risk?
Kind of, yeah. They used ApoE knockout mice. They injected pristine C60 into the abdomen and checked aorta function just an hour later.
Quick effects.
Yeah.
And what happened in these atherosclerosis-prone mice?
The main takeaway was C60 treatment led to reduced vasorelaxation. Their main artery, the aorta, couldn't relax as well as it should.
So again, an effect on blood vessel function, making them sort of stiffer, less
Well,
responsive.
Less able to relax.
Yeah.
Specifically, the relaxation that depends on the endothelium, the inner lining. This was seen in female mice, both young and old. There was also a slight reduction in relaxation that doesn't depend on the endothelium, but mainly in young mice at a higher dose.
Did it affect constriction?
Not really, and no significant changes there. And they did describe the relaxation effect as modest.
Modest, okay. Not a huge effect, but measurable.
Right. And because it happened so fast, just 1 hour, they figured it probably wasn't inflammation. Maybe, they suggested, direct oxidative stress generated by C60 in the blood vessel wall itself, which implies the C60 got there pretty quickly.
And did the study also highlight complexity?
Oh, absolutely. They made a point that different types of C60, the basic pristine kind they used versus ones that have been chemically modified, can act very differently on blood vessels.
So you can't just assume all C60 is the same.
Not at all. And dose mattered too. Sometimes the lower dose had more effect than the higher dose in their study. It's not straightforward.
Okay, let's try and synthesize this. We've got the body rat study showing this incredible, unexpected longevity boost. We've got a personal report of feeling benefits like energy and focus.
Potential upsides.
But then we have these other lab studies. One showing C60 worsening heart damage in rats under specific stress conditions. Another showing it can act as a potent oxidant, especially with light in zebrafish. And a third showing modest negative effects on blood vessel relaxation in vulnerable mice.
Exactly. It's this contrast that's so striking when you look at all the sources together. Hints of benefit alongside documented potential for harm or complex interactions for controlled settings.
And it really hammers home those key variables you mentioned that seem to influence everything.
Yeah, it seems critical how the C60 gets into the system. Lungs, injection, orally makes a difference. The type of C60, basic versus modified, clearly matters.
Environmental factors like light, maybe.
Appears so, based on the zebrafish. And the biological context, the organism's own antioxidant levels, its sex, whether it has pre-existing conditions like atherosclerosis, all seem to modulate the response.
So for you listening, if you're hearing the buzz about C60, maybe seeing it sold as a supplement, these sources really highlight how tangled the science is. You've got to consider the source of the info, that one eye-popping study, a personal story, or these targeted lab studies.
Right. And understand that C60's effects aren't simple. They seem highly dependent on all these factors, the form it's in, how you're exposed, your own biology.
Which loops back to what the blog author said about quality. If the form and purity matter so much,
then transparency and testing become paramount. Knowing exactly what you're getting, how pure it is, how it's prepared, that seems absolutely essential given the variability seen in these studies.
So what does this all mean in the end? Where does this leave us?
Well, I think it means C60 is a classic example of something incredibly interesting scientifically, with hints of real potential, but also clear signals for caution based on how it behaves in different lab models.
From that stunning longevity finding and the user report,
To the concerning cardiovascular findings, the oxidant potential, the blood vessel effects, It's a mixed bag.
And the science is clearly still young. Figuring out how rat, zebrafish, or mouse studies translate to humans taking C60 oil, that's a huge gap
A
still.
massive gap. So based on these sources, C60 isn't a simple thumbs up or thumbs down. Its effects look really contingent on form, delivery, light, biology. Context is everything.
Which leaves us with a final thought, maybe for you to ponder. Given everything we've seen, the conflicting results, how much things like the exact chemical form or exposure route seem to matter.
Yeah.
How should we approach evaluating these kinds of nanomaterials like C60 as they potentially move from specialized labs towards consumer products or even medical use, especially when the picture is this complex?
That's a really important question, isn't it? How do we navigate that path from discovery to safe application when the variables are so numerous and the effects potentially so context dependent? Something to think of.